Actually, before we get into the structure of the nucleosomes, I want to talk about different types of chromatin. So you can have euchromatin or you can have heterochromatin. And they're most easily seen in a polytene chromosome. These are often taken from salivary glands in Drosophila larvae. And what happens in these salivary glands is that chromosomes replicate many times without any cell division. And so what you have is this polytene chromosome structure, which is made up of many copies of the same chromosome. And so you can actually see through the microscope regions that are darker, that represent areas that are what we call heterochromatic, and regions that are lighter, they also can be called chromosome puffs, where the chromosome structure is looser, and these are euchromatic regions. And the euchromatic regions are where transcription is actively happening, and the heterochromatic regions that are darker are the regions that are transcriptionally silent. Okay, so two types of chromatin, heterochromatin and euchromatin. And the difference between heterochromatin and euchromatin is largely around how the DNA is packaged by nucleosomes. And so when nucleosomes are closer together, they are packaged more tightly, the DNA is packaged more tightly, and you get heterochromatin. When there's more space between the nucleosomes, the DNA packaging is looser, and you allow transcription factors and other proteins that are necessary for transcription to access the DNA. So these nucleosomes are comprised of histones, 8 total. So there's 4 types of histones. There's 2A, 2B, 3, and 4. And there are 4 types of each of these, or sorry, 2 of each. So there's a total of 8 histone proteins within the nucleosome that are 2 copies each of 4 types of histones, 2A, 2B, 3, and 4. Now, there are also these histone tails that extend past the nucleosome. So the nucleosome in general includes both the histone and the DNA. The histone tails extend past the nucleosome, and they're actually, let's see what the next slide, okay, so they're actually what helps create a tighter or a looser bond of the histones with the DNA. So the histone tails are naturally more positively charged, which means that they are attracted to the negative charge of the DNA. They hold on more tightly, and in that case, they're normally they're going to be complexing with the DNA fairly tightly, and that creates a more heterochromatic structure. When you have so on histone tails, there are a number of acetyl groups. And when you deacetylate these structures on the chromatin tails, you actually make them more negatively charged. So specifically, histone tails are fairly rich with lysines, and these lysines can have acetyl groups added to them. And these are done by acetyltransferase enzymes. And so let's say here's a lysine on a histone tail. You suddenly have an acetyl group added to it, and that makes it more negatively charged. And so it ends up letting go of the DNA a little bit more, so it's not so tightly bound. And so what happens is nucleosomes are actually able to slide up and down the DNA and adjust their positions based on a number of protein interactions, as well as what we call histone modifications, particularly of the histone tails, that influence the way the tails are interacting with the DNA. Okay, so these types of modifications are often called epigenetic modifications because they are inherited through cell divisions. And so when these histone changes happen within a cell, when that cell divides, the daughter cells will also have those histone changes. If this happens within the germline, then the histone changes that happen in the parent can also be transferred to the offspring. So these can have huge phenotypic effects. One example is the agouti locus in mice causes yellow coloration. It actually influences where black pigment is placed on fur. And when it's silenced, and this silencing is through epigenetic modifications, you get a yellow mouse that's kind of prone to obesity. When the agouti is allowed to be expressed, then you get a brown mouse that is not. And so these mice are all genotypically identical, but they vary in the degree at the agouti locus, but they vary to the degree to which that locus is silenced. Okay? And so you have an extra layer then of genetic influence where it's not just your genotype, whether you have the agouti allele or not, but whether or not that allele is allowed to actually be expressed through, in this case, through epigenetic modifications. Okay, here's a clicker question, just food for thought here. Reducing the positive charge of histone proteins would have which effect on them? A, they would bind the DNA tighter, they would bind less tightly to the DNA, they would no longer be attracted to each other, or they would cause supercoiling of the DNA. I'm going to give you a few minutes to think that over. So because DNA is negatively charged, if you reduce the positive charge of histone proteins and actually make them more negatively charged, they're going to be binding less tightly to the DNA. Here's another one. How many copies of the H2B histone would be found in a chromatin containing 50 nucleosomes? You can pause it here too and just kind of, yeah. So because each nucleosome contains 2 copies of every type of histone, we would have 2 times 50, which is 100 nucleosomes. Okay, so moving on to larger chromosome structure. We talked about this before, where chromosomes are characterized by having a centromere somewhere in it, or a and telomeres at the end. Specifically here we're talking about eukaryotic linear chromosomes. Here we have one that's been replicated. It has 2 sister chromatids. Each chromatid has its own telomere and a single centromeric region. So just a little word on centromeres. Centromeres are characterized by repetitive DNA, and what that means is that the DNA is characterized by lots of repeats. There generally aren't a lot of genes in the centromere, and it tends to be a transcriptionally silent region. Centromeres, as we know, can be located anywhere along the chromosome, and then in some species, chromosomes can have multiple centromeres, or sort of regions that are larger centromeric regions. So the centromere in and of itself is kind of difficult to define. We know that in many cases it's the site of attachment for the spindle apparatus during cell division, but in other species that doesn't seem to be necessary, and we're not going to talk about that. But I want you to be aware that there is quite a bit of variation when it comes to centromere structure. But for now, we're going to be, when we think about centromeres, we're going to be thinking about a single location somewhere on the chromosome that is highly repetitive in DNA sequence, tends to be transcriptionally silent, and is a region where spindle fibers attach during metaphase and anaphase. Okay, So telomeres are kind of cool because they have characteristics. They're also repetitive, and they have characteristic sequences. And the sequence can be described as being A/T to the N, G to the M. With let me just double check this. So the way that I have it in the notes is that it's A/T to the M and then G to the N. What this means is that there's a certain number of A's and T's, A's or T's. There's a region that's AT rich, followed by a region that has G's, right? And so the sequence of the telomere repeat varies for different groups of species or taxa. And but it is fairly predictable in that these sequences are all fairly similar in their pattern. So again, telomeres protect the ends of DNA. Normally when DNA is broken, it degrades very easily, and telomeres protect the degradation at the ends of the DNA. So there's a G-rich strand. So there's a strand that has the A's and T's and then is C-rich. There's a strand that's G-rich. And the strand that's G-rich tends to produce this 3' overhang. And sometimes there's a multi-protein complex that binds to telomeres to kind of prevent degradation of this end. But other times the 3' overhang may bind with an upstream sequence. So upstream meaning in this direction, it may come around and bind with a sequence. So here's our 3' overhead, binding with an upstream sequence and forming what we call a T-loop. And it's this T-loop that helps to create, suddenly now we no longer have a very visible end to the chromosome, and this is what protects it. Okay, so we're gonna, the extension or the maintenance of the telomere is done by telomerase, and we'll talk about exactly how that works in another lecture. Alright, so I mentioned that DNA is repetitive. There's a number of different ways that DNA can be characterized. There's fairly unique sequence DNA that isn't very repetitive, and then there's a kind of a continuum between from totally unique DNA sequence that's found nowhere else in the genome to highly repetitive DNA that is just a similar sequence repeated over and over and over again. And I just want to kind of, this will be kind of a recurring theme, the theme of DNA repetitiveness. So we saw it first in the centromere. We're seeing it again in the telomeres, and it's going to kind of come up again and again. And different genomes of different species have different degrees of repetitiveness. Our DNA, human DNA, is highly, highly, highly repetitive and has long intergenic distances filled with repetitive DNA. Genomes of other organisms, including Drosophila, are somewhat less repetitive, but at the same time there's differences in repetitiveness among different species as well. Here's just a clicker question. What is characteristic of DNA sequences at the telomeres? A, one strand consists of guanine and adenine or thymine nucleotides. B, they consist of repeated sequences. C, one strand protrudes beyond the other, creating some single-stranded DNA at the end. Or D, all of the above. So the correct answer is all of the above, because all of those things are true.